Methods for genetic control of insect infestations in plants and compositions. Vera VP 2431473A1 Abstract. The present invention relates to control of pest infestation by inhibiting one or more biological functions. The invention provides methods and compositions for such control by feeding one or more recombinant double-stranded RNA molecules provided by the invention to the pest. The reduction in pest infestation is obtained through suppression of gene expression. The invention is also directed to methods for making transgenic plants that express the double-stranded RNA molecules, and to particular combinations of transgenic pesticidal agents for use in protecting plants from pest infestation. Images 10 Patent Drawing Search Report 1 Patent Drawing Search Report 2 Patent Drawing Search Report 3 Patent Drawing Patent Drawing Patent Drawing Patent Drawing Patent Drawing Patent Drawing Patent Drawing. Next page. Claims 18. An isolated polynucleotide selected from a group consisting of a polynucleotide comprising a nucleic acid sequence of seqid no: 818. B. A polynucleotide that hybridizes to a nucleic acid sequence of seqid no: 818 under wash conditions of 5xSSC, 50% formaldehyde and 42 degrees C for 10 minutes. C. A polynucleotide having at least 70% sequence identity to a nucleic acid sequence of seqid no: 818. D. A fragment of at least 21 contiguous nucleotides of a nucleic acid sequence of seqid no: 818, and E. A complement of the sequence of ABC or D wherein ingestion by a coleopteran plant pest of a double-stranded ribonucleotide sequence comprising at least one strand that is complementary to said polynucleotide or said fragment inhibits the growth of said pest. The isolated polynucleotide of Claim 1, which I is defined as operatively linked to a heterologous promoter, or II is defined as comprised on a plant transformation vector. The double-stranded ribonucleotide sequence produced from the expression of a polynucleotide according to Claim 1, wherein ingestion of said ribonucleotide sequence by a coleopteran plant pest inhibits the growth of said pest. The double-stranded ribonucleotide sequence of Claim 3, I defined as produced by preparing the recombinant polynucleotide sequence comprising a first, a second and a third polynucleotide sequence, wherein the first polynucleotide sequence comprises the isolated polynucleotide of Claim 1, wherein the third polynucleotide sequence is linked to the first polynucleotide sequence by the second polynucleotide sequence, and wherein the third polynucleotide sequence is substantially the reverse complement of the first polynucleotide sequence such that the first and the third polynucleotide sequences hybridize when transcribed into a ribonucleic acid to form the double-stranded ribonucleotide molecule stabilized by the linked second ribonucleotide sequence, or II, wherein ingestion of the polynucleotide sequence by the pest inhibits the expression of a nucleotide sequence substantially complementary to said polynucleotide sequence. The cell transformed with the polynucleotide of Claim 1. The cell of Claim 5, defined as I a prokaryotic cell, or II a eukaryotic cell, or II a plant or bacterial cell. The plant transformed with the polynucleotide of Claim 1 or 2, or a seed thereof comprising the polynucleotide. The plant of Claim 7, wherein said polynucleotide is expressed in a cell of the plant as a double-stranded ribonucleotide sequence and ingestion of an insect pest inhibitory amount of said double-stranded ribonucleotide sequence in a diet inhibits the pest from further feeding on said diet, preferably I. The insect pest is selected from Diabrotica virgifera, Diabrotica virgifera virgifera, Diabrotica virgiferazea, Diabrotica baltirda, Diabrotica barbaris, Diabrotica virgula, Diabrotica speciosa, and Diabrotica undecimpunctatus, or II. Ingestion of the insect pest inhibitory amount of a double-stranded ribonucleotide sequence stunts the growth of the pest. A commodity product produced from a plant according to Claim 7, wherein said commodity product comprises a detectable amount of the polynucleotide of Claim 1 or a ribonucleotide expressed therefrom. The method for controlling coleopteran pest infestation comprising providing in the diet of a coleopteran pest an agent comprising a first polynucleotide sequence that functions upon ingestion by the pest to inhibit biological function within said pest, wherein said polynucleotide sequence exhibits from about 95 to about 100% nucleotide sequence identity along at least from about 19 to about 25 contiguous nucleotides to a coding sequence derived from said pest and is hybridized to a second polynucleotide sequence that is complementary to said first polynucleotide sequence, and wherein said coding sequence derived from said pest is SEQIDN 0:818, or the complement thereof. The method of Claim 10, wherein said Coleopteran pest is a Diabrotica spp, which preferably is selected from Diabrotica virgifera, Diabrotica virgifera, Diabrotica virgiferazea, Diabrotica bultira, Diabrotica barbari, Diabrotica virgula, Diabrotica speciosa, and Diabrotica undecimpunctata. The method for controlling the Coleopteran pest infestation comprising providing in the diet of a Coleopteran pest a plant cell expressing a polynucleotide sequence according to Claim 1, wherein the polynucleotide is expressed to produce a double-stranded ribonucleic acid that functions upon ingestion by the pest to inhibit the expression of a target sequence within said pest and results in decreased feeding on said diet relative to a diet lacking the plant cell. The method of Claim 12, wherein by the pest exhibits reduced growth following ingestion of the cell, or II the plant cell further comprises a polynucleotide sequence encoding a pesticidal agent selected from a potaton, the Bacillus thuringiensis insecticidal protein, as Enterhabdus insecticidal protein, the Photorhabdus insecticidal protein, the Bacillus laterosporus insecticidal protein, and a Bacillus sphaericus insecticidal protein. Preferably said Bacillus thuringiensis insecticidal protein is selected from a Cry1, the Cry3, a Tick 851, a Cryat 70, the Cry22, a Tick 901, a Tick 201, a Tick 407, a Tick 417, the binary insecticidal protein Cryat 33 and Cryat 34, the binary insecticidal protein Cryat 80 and Cryat 76. The binary insecticidal protein VIC-100 and VIC-101, the combination of the insecticidal proteins at 29 or at 37 with insecticidal proteins TIC-810 or TIC-812, and a binary insecticidal protein PS-149B1, or II the target sequence encode the protein, the predicted function of which is selected from muscle formation, juvenile hormone formation, juvenile hormone regulation, ion regulation and transport, digestive enzyme synthesis, maintenance of cell membrane potential, amino acid biosynthesis, amino acid degradation, sperm formation, pheromone synthesis, pheromone sensing, and any formation, wing formation, leg formation, development and differentiation, egg formation, larval maturation, digestive enzyme formation, hemolymph synthesis, hemolymph maintenance, neurotransmission, cell division, energy, metabolism, respiration, and apoptosis. More IV said Coleopteran pest is a Diabrotica spp, which preferably is selected from Diabrotica virgifera, Diabrotica virgifera, Diabrotica virgiferazea, Diabrotica bultira, Diabrotica barbari, Diabrotica virgula, Diabrotica speciosa, and Diabrotica undecimpunctata, or V. The polynucleotide functions upon ingestion by the pest to suppress a gene that performs a function essential for insect survival, said function being selected from feeding by the pest, pest cell apoptosis, cell differentiation and development, capacity or desire for sexual reproduction, muscle formation, muscle twitching, muscle contraction, juvenile hormone formation, juvenile hormone regulation, ion regulation and transport, maintenance of cell membrane potential, amino acid biosynthesis, amino acid degradation, sperm formation, pheromone synthesis, pheromone sensing, and any formation, wing formation, leg formation, egg formation, larval maturation, digestive enzyme formation, hemolymph synthesis, hemolymph maintenance, neurotransmission, larval stage transition, pupation, emergence from pupation, cell division, energy metabolism, respiration, and formation of cytoskeletal structure. The method for improving the yield of a crop produced from a crop plant subjected to insect pest infestation. Said method comprising the steps of A. Introducing a polynucleotide according to Claim 1 into said crop plant. B. Cultivating the crop plant to allow the expression of said polynucleotide, wherein expression of the polynucleotide inhibits feeding by insects, pests, and loss of yield due to pest infestation. The method of Claim 14, wherein by expression of the polynucleotide produces an RNA molecule that suppresses at least a first target gene in an insect pest that has ingested a portion of said crop plant, wherein the target gene performs at least one essential function selected from feeding by the pest. Viability of the pest. Pest cell apoptosis. Differentiation and development of the pest or any pest cell. Sexual reproduction by the pest. Muscle formation, muscle twitching, muscle contraction, juvenile hormone formation and / or reduction, juvenile hormone regulation, ion regulation and transport, maintenance of cell membrane potential, amino acid biosynthesis, amino acid degradation, sperm formation, pheromone synthesis, pheromone sensing, and any formation, wing formation, leg formation, egg formation, larval maturation, digestive enzyme formation, hemolymph synthesis, hemolymph maintenance, neurotransmission, larval stage transition, pupation, emergence from pupation, cell division, energy metabolism, respiration, cytoskeletal structure synthesis and maintenance, nucleotide metabolism, nitrogen metabolism, water use, water retention, and sensory perception, or IID. Insect pest is a corn rootworm pest selected from Diabrotica undecimpunctata howardi. Southern corn rootworm SCR Diabrotica virgifera western corn rootworm WCR Diabrotica barbari. Northern corn rootworm NCR Diabrotica virgifera Mexican corn rootworm MCR Diabrotica baltira. Brazilian corn rootworm BZR Diabrotica virgula. Brazilian corn rootworm BZR and Diabrotica species of Brazilian corn rootworm BZR. The method for improving the drought tolerance of a crop produced from a crop plant subjected to insect pest infestation. Said method comprising the steps of A. Introducing a polynucleotide according to Claim 1 into said crop plant. B. Cultivating the crop plant to allow the expression of said polynucleotide. Where an expression of the polynucleotide inhibits feeding by insects, pests, and loss of drought tolerance due to pest infestation. The method of producing a commodity product, food or feed comprising obtaining a plant according to Claim 7 or part thereof, and preparing a commodity product, food or feed from the plant or part thereof. The method of Claim 17, wherein the food or feed is defined as oil, meal, protein, starch, flour or silage. Description incorporation by reference of sequence listing submitted on a compact disc 0001. The sequence listing is submitted on one compact disc copy 1 together with a duplicate Vera copy 2, each created on September 15, 2006, and each containing one 669 kilobytes file entitled mde002.app.txt. The material contained on the compact disc is specifically incorporated herein by reference. Priority Claim 0002. This application claims the priority of U.S. Provisional Application SEER. Number 60/718,034, filed September 16, 2005, which application is incorporated herein by reference in its entirety. Background of the invention 1. Field of the invention 0003. The present invention relates generally to genetic control of pest infestations. More specifically, the present invention relates to recombinant DNA technologies for post-transcriptionally repressing or inhibiting expression of target coding sequences in the cell of a pest to provide a pest protective effect. 2. Description of related R000 for the environment in which humans' life is replete with pest infestation. Pests including insects, arachnids, crustaceans, fungi, bacteria, viruses, nematodes, flatworms, roundworms, pinworms, hookworms, tapeworms, trypanosomes, schistosomes, botflies, fleas, ticks, mites, and lice and the like are pervasive in the human environment. A multitude of means have been utilized for attempting to control infestations by these pests. Compositions for controlling infestations by microscopic pests such as bacteria, fungi, and viruses have been provided in the form of antibiotic compositions, antiviral compositions, and antifungal compositions. Compositions for controlling infestations by larger pests such as nematodes, flatworm, roundworms, pinworms, heartworms, tapeworms, trypanosomes, schistosomes, and the like have typically been in the form of chemical compositions that can be applied to surfaces on which pests are present or administered to infested animals in the form of pellets, powders, tablets, pastes, or capsules and the like. There is a great need in the art for improvement of these methods, and particularly for methods that would benefit the environment relative to the prior techniques. 0005 Commercial crops are often the targets of insect attack. Substantial progress has been made in the last few decades towards developing more efficient methods and compositions for controlling insect infestations in plants. Chemical pesticides have been very effective in eradicating pest infestations. However, there are several disadvantages to using chemical pesticidal agents. Chemical pesticidal agents are not selective. Applications of chemical pesticides intended to control invertebrate pests, such as coleoptera insects including corn rootworm species that are harmful to various crops and other plants, exert their effects on non-target fauna as well, often effectively sterilizing the field for a period of time over which the pesticidal agents have been applied. Chemical pesticidal agents persist in the environment and generally are slow to be metabolized, if at all. They accumulate in the food chain, and particularly in the higher predator species. Accumulations of these chemical pesticidal agents results in the development of resistance to the agents, and in species higher up the evolutionary ladder, can act as mutagens and or carcinogens to cause irreversible and deleterious genetic modifications. Thus there has been a particularly long-felt need for environmentally friendly methods for controlling or eradicating insect infestation on or in plants, i.e., methods that are selective, environmentally inert, non-persistent, and biodegradable, and that fit well into pest resistance management schemes. 0006 Compositions that include Bacillus thuringiensis BD bacteria have been commercially available and used as environmentally safe and acceptable insecticides for more than 30 years. The insecticidal effect of BD bacteria do not persist in the environment, are highly selective as to the target species affected, exert their effects only upon ingestion by a target pest, and have been shown to be harmless to plants and other non-targeted organisms, including humans. Transgenic plants containing one or more genes encoding insecticidal BD protein are also available in the R and are remarkably efficient in controlling insect pest infestation. A substantial result of the use of recombinant plants expressing BD insecticidal proteins is a marked decrease in the amount of chemical pesticidal agents that are applied to the environment to control pest infestation in crop fields in areas in which such transgenic crops are used. The decrease in application of chemical pesticidal agents has resulted in cleaner soils and cleaner waters running off of the soils into the surrounding streams, rivers, ponds and lakes. In addition to these environmental benefits, there has been a noticeable increase in the numbers of beneficial insects in crop fields in which transgenic insect-resistant crops are grown because of the decrease in the use of chemical insecticidal agents. 0007 Antisense methods and compositions have been reported in the R and are believed to exert their effects through the synthesis of a single-stranded RNA molecule that in theory hybridizes in vivo to a substantially complementary sense-strand RNA molecule. Antisense technology has been difficult to employ in many systems for three principal reasons. First, the antisense sequence expressed in the transformed cell is unstable. Second, the instability of the antisense sequence expressed in the transformed cell concomitantly creates difficulty in delivery of the sequence to a host, cell type, or biological system remote from the transgenic cell. Third, the difficulties encountered with instability and delivery of the antisense sequence create difficulties in attempting to provide a dose within the recombinant cell expressing the antisense sequence that can effectively modulate the level of expression of the target sense nucleotide sequence. 0008 There have been few improvements in technologies for modulating the level of gene expression within a cell, tissue, or organism, and in particular, the lack of developed technologies for delaying, repressing or otherwise reducing the expression of specific genes using recombinant DNA technology. Furthermore, as a consequence of the unpredictability of these approaches, no commercially viable means for modulating the level of expression of a specific gene in a eukaryotic or prokaryotic organism is available. 0009 Double-stranded RNA-mediated inhibition of specific genes in various pests has been previously demonstrated. These RNA-mediated approaches to genetic control have been tested in the fruit fly Drosophila melanogastric Enerdell and Carthusia, 1998, Enerdell and Carthusia, 2000. Kennardell and Carthouse 1998 describe a method for delivery of dsRNA involved generating transgenic insects that express double-stranded RNA molecules or injecting dsRNA solutions into the insect body or within the egg sac prior to or during embryonic development. 0010 Research investigators have previously demonstrated that double-stranded RNA-mediated gene suppression can be achieved in nematodes either by feeding or by soaking the nematodes in solutions containing double-stranded or small interfering RNA molecules and by injection of the dsRNA molecules. Rahagopal al. 2002 describe failed attempts to suppress an endogenous gene in larvae of the insect pest Pedoptera by feeding or by soaking neonate larvae in solutions containing dsRNA specific for the target gene, but were successful in suppression after larvae were injected with dsRNA into the hemolymph of fifth instar larvae using a microapplicator. Recently, Adad et al. 2006 reported that host-generated dsRNA produced in a plant can protect such plants from infection by nematodes. Similarly, U.S. Patent APP Pub. Number 2003/01500017 prophetically described a preferred locus for inhibition of the Lepidopteran larvae Helicoverpa armatura using dsRNA delivered to the larvae by ingestion of a plant transformed to produce the dsRNA. 2005/110068 teaches providing, in the diet of corn rootworm CRW specific dsRNA directed to essential CRW genes. The dsRNA is provided in the CRW diet in vitro and in planta, with the result that CRW larvae are stunted or killed after feeding on the diet, and this effect was demonstrated for several different genes. 0011 Therefore, there has existed a need for identifying efficacious nucleotide sequences for use in improved methods of modulating gene expression by repressing, delaying or otherwise reducing gene expression within a particular coleopteran pest for the purpose of controlling pest infestation or to introduce novel phenotypic traits. Summary of the invention 0012 in one aspect. The invention provides a method of inhibiting expression of a target gene in a coleopteran pest. In certain embodiments, the method comprises modulating or inhibiting expression of one or more target genes in a coleopteran pest that causes cessation of feeding, growth, development, reproduction and/or infectivity and eventually result in the death of the insect. The method comprises introduction of partial or fully stabilized double-stranded RNA dsRNA including its modified forms such as small interfering RNA siRNA sequences, into the cells or into the extracellular environment, such as the midgut, within a coleopteran pest body wherein the dsRNA enters the cells and inhibits expression of at least one or more target genes and wherein the inhibition exerts a deleterious effect upon the coleopteran pest. The methods and associated compositions may be used for limiting or eliminating coleopteran pest infestation in or on any pest host, pest symbiont, or environment in which a pest is present by providing one or more compositions comprising the dsRNA molecules described herein in the diet of the pest. The method will find particular benefit for protecting plants from insect attack. In one embodiment, the pest is defined as comprising the digestive system pH within a range of from about 4.5 to about 9.5, from about 5 to about 9, from about 6 to about 8, and from about pH 7.0. 0013 In another aspect, the present invention provides exemplary nucleic acid compositions that are homologous to at least a portion of one or more native nucleic acid sequences in a target pest. In certain embodiments, the pest is selected from among Diabrotica sp, including western corn rootworm WCR, Diabrotica virgifera or Diabrotica virgifera southern corn rootworm SCR, Diabrotica undecimpunctata howardi Mexican corn rootworm MCR, Diabrotica virgifera a Brazilian corn rootworm PZR, Diabrotica bultura, Diabrotica virgula, Diabrotica species a northern corn rootworm NCR, Diabrotica barberi Diabrotica undecimpunctata, as well as Colorado potato beetle CPB, Leptinotarsa disseminata red flower beetle RFB, Tribolium castaneum and Mexican bean beetle Epilochna viridis. In other embodiments the pest is selected from among lepidopteran insects including European corn borer BCB, Australia nubilalis black cutworm BCW, Agrodes ypsilon corn earworm CEW, Helicoverpa fall armyworm fawn. Sphagnoptera frugivora cotton ball weevil BWV, Anthonymous grandis silkworms Bombyx mori and Mangicus sexta, and from dectrin insects including Drosophila melanogaster, Anopheles gambiae, and Anthes aegypti. Specific examples of such nucleic acids provided by the invention are given in the attached sequence listing as SEQID No:1 through SEQID No:906. 001 For yet another aspect, the invention provides a method for suppression of gene expression in a coleopteran pest such as a corn rootworm or related species that comprises the step of providing in the diet of the pest the gene suppressive amount of at least one dsRNA molecule transcribed from a nucleotide sequence as described herein, at least one segment of which is complementary to an mRNA sequence within the cells of the pest. The method may further comprise observing the death, inhibition, stunting, or cessation of feeding of the pest. A dsRNA molecule, including its modified form such as an siRNA molecule, federal to a pest in accordance with the invention may be at least from about 80, 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, or about 100%. Identical to a RNA molecule transcribed from a nucleotide sequence selected from the group consisting of SEQID No:1 through SEQID No:906. In particular embodiments, the nucleotide sequence may be selected from the group consisting of SEQID No:697, SEQID NOS:813-819, SEQID No:841, and SEQID No:874. 0015 Accordingly, in another aspect of the present invention, a set of isolated and purified nucleotide sequences as set forth in SEQID No:1 through SEQID No:906 is provided. The present invention provides a stabilized dsRNA molecule for the expression of one or more mRNAs for inhibition of expression of a target gene in a coleopteran pest expressed from these sequences and fragments thereof. The stabilized dsRNA, including the mRNA or siRNA molecule, can comprise at least two coding sequences that are arranged in a sense and an antisense orientation relative to at least one promoter, wherein the nucleotide sequence that comprises a sense strand and an antisense strand are linked or connected by a spacer sequence of at least from about 5 to about 1000 nucleotides, wherein the sense strand and the antisense strand may be a different length, and wherein each of the two coding sequences shares at least 80% sequence identity, at least 90%, at least 95%, at least 98%, or 100 degrees/O sequence identity, to any one or more nucleotide sequence as set forth in SEQID No:1 through SEQID No:906. 0016 Further provided by the invention is a fragment or concatemer of a nucleic acid sequence selected from the group consisting of SEQID No:1 through SEQID No:906. In particular embodiments, the nucleotide sequence may comprise a fragment or concatemer of a sequence selected from the group consisting of SEQID No:697, SEQID NOS:813-819, SEQID No:841, and SEQID No:874. 0017 The fragment may be defined as causing of the death, inhibition, stunting, or cessation of feeding of a pest when expressed as a dsRNA and provided to the pest. The fragment may, for example, comprise at least about 19, 21, 23, 25, 40, 60, 80, 100, 125 or more contiguous nucleotides of any one or more of the sequences in SEQID No:1 through SEQID No:906, or a complement thereof. One beneficial DNA segment for use in the present invention is at least from about 19 to about 23, or about 23 to about 100 nucleotides up to about 2000 nucleotides or more in length. Particularly useful will be dsRNA sequences including about 23 to about 300 nucleotides homologous to a pest target sequence. The invention also provides a ribonucleic acid expressed from any of such sequences including a dsRNA. A sequence selected for use in expression of a gene suppression agent can be constructed from a single sequence derived from one or more target pests and intended for use in expression of an RNA that functions in the suppression of a single gene or gene family in the one or more target pests, or that the DNA sequence can be constructed as a chimera from a plurality of DNA sequences. 0018 In yet another aspect, the invention provides recombinant DNA constructs comprising a nucleic acid molecule encoding a dsRNA molecule described herein. The dsRNA may be formed by transcription of one strand of the dsRNA molecule from a nucleotide sequence which is at least from about 80% to about 100% identical to a nucleotide sequence selected from the group consisting of seqid No through seqid No: 906. Such recombinant DNA constructs may be defined as producing dsRNA molecules capable of inhibiting the expression of endogenous target gene S in a pest cell upon ingestion. The construct may comprise the nucleotide sequence of the invention operably linked to a promoter sequence that functions in the host cell. Such a promoter may be tissue-specific and may, for example, be specific to a tissue type which is the subject of pest attack. In the case of rootworms, for example, it may be desired to use a promoter providing root-preferred expression. 0019 Nucleic acid constructs in accordance with the invention may comprise at least one non-naturally occurrent nucleotide sequence that can be transcribed into a single-stranded RNA capable of forming a dsRNA molecule in vivo through hybridization. Such dsRNA sequences self-assemble and can be provided in the diet of a coleopteran pest to achieve the desired inhibition. 0020 A recombinant DNA construct may comprise two different non-naturally occurrent sequences which, when expressed in vivo as dsRNA sequences and provided in the diet of a coleopteran pest, inhibit the expression of at least two different target genes in the cell of the coleopteran pest. In certain embodiments, at least 3, 4, 5, 6, 8 or 10 or more different dsRNAs are produced in a cell or plant comprising the cell that have a pest inhibitory effect. The dsRNAs may be expressed from multiple constructs introduced in different transformation events or could be introduced on a single nucleic acid molecule. The dsRNAs may be expressed using a single promoter or multiple promoters. In one embodiment of the invention, single dsRNAs are produced that comprise nucleic acids homologous to multiple loci within a pest. 0021 In still yet another aspect, the invention provides a recombinant host cell having in its genome at least one recombinant DNA sequence that is transcribed to produce at least one dsRNA molecule that functions when ingested by a coleopteran pest to inhibit the expression of a target gene in the pest. The dsRNA molecule may be encoded by any of the nucleic acids described herein and as set forth in the sequence listing. The present invention also provides a transformed plant cell having in its genome at least one recombinant DNA sequence described herein. Transgenic plants comprising such a transformed plant cell are also provided, including progeny plants of any generation, seeds, and plant products, each comprising the recombinant DNA. 0022 The methods and compositions of the present invention may be applied to any monocot and dicot plant, depending on the coleopteran pest control desired. Specifically, the plants are intended to include, without limitation, alfalfa, peanut, apple, apricot, artichoke, arugula, asparagus, avocado, bananas, barley, beans, beet, blackberry, blueberry, broccoli, Brussels sprouts, cabbage, canola, cantaloupe, carrot, cassava, cauliflower, celery, cherry, cilantro, citrus, clementine, coffee, corn, cotton, cucumber, Douglas fir, eggplant, endive, escarole, eucalyptus, fennel, figs, gourd, grape, grapefruit, honeydew, jicama, kiwi fruit, lettuce, leeks, lemon, lime, loblolly pine, mango, melon, mushroom, nut, oat, okra, onion, orange, an ornamental plant, papaya, parsley, pea, peach, peanut, pear, pepper, persimmon, pine, pineapple, plantain, plum, pomegranate, poplar, potato, pumpkin, quince, radiata pine, radicchio oak, radish, raspberry, rice, right, sorghum, southern pine, soybean, spinach, squash, strawberry, sugar beet, sugarcane, sunflower, sweet potato, sweet gum, tangerine, tea, tobacco, tomato, turf, a vine, watermelon, wheat, yams, and zucchini plants. Thus, the plant transformed with a recombinant DNA sequence as set forth in SEQID No. through SEQID No: 906, or concatemer, fragment, or complement thereof, that is transcribed to produce at least one dsRNA molecule that functions when ingested by a coleopteran pest to inhibit the expression of a target gene in the pest is also provided by the invention. In particular embodiments, the recombinant DNA sequence may be selected from the group consisting of SEQID No: 697, SEQID NOS: 813-819, SEQID No: 841, and SEQID No: 874, or fragment, complement, or concatemer thereof. 0023 The invention also provides combinations of methods and compositions for controlling coleopteran pest infestations. One means provides a dsRNA method as described herein for protecting plants from insect infestation along with one or more insecticidal agents that exhibit features different from those exhibited by the dsRNA methods and compositions. For example, one or more Bt proteins may be provided in the diet of insect pests in combination with one or more dsRNAs as described herein. A composition formulated for topical application or derived using a transgenic approach that combines dsRNA methods and compositions with Bt may be used to provide synergies that were not known previously in the art for controlling insect infestation. One synergy is the reduction in the level of expression required for either the dsRNA or for the Bt protein S. When combined together, the lower effective dose of each pest control agent could be used. It is believed that the Bt insecticidal proteins create entry ports through which the dsRNA molecules are able to penetrate more effectively into spaces remote from the gut of the insect pest, or more efficiently into the cells in the proximity of lesions created by the Bt proteins, thus requiring less of either the Bt or the dsRNA to achieve the desired insecticidal result or the desired inhibition or suppression of a targeted biological function in the target pest. 0024 The present invention therefore provides a composition that contains two or more different pesticidal agents each toxic to the same pest or insect species, at least one of which comprises a dsRNA described herein. In certain embodiments, the second agent can be an agent selected from the group consisting of a potaton, the Bacillus thuringiensis insecticidal protein, a Xenarhabdus insecticidal protein, the Photorhabdus insecticidal protein, the Bacillus laterosporus insecticidal protein, the Bacillus sphaericus insecticidal protein, and a lignin. The Bacillus thuringiensis insecticidal protein can be any of a number of insecticidal proteins including but not limited to a Cry1, the Cry3, a TIC851, a Cryat70, the Cry22, a TIC901, a TIC1201, a TIC407, a TIC417, the binary insecticidal protein Cryat33 and Cryat34, the binary insecticidal protein Cryat80 and Cryat76, the binary insecticidal protein TIC100 and TIC101, the binary insecticidal protein PS149B1, a VIP insecticidal protein, a TIC900 or related protein, or combinations of the insecticidal proteins at 29 or at 37 with insecticidal proteins TIC810 or TIC812, and insecticidal chimeras of any of the preceding insecticidal proteins. 0025 The ribonucleic acid that is provided in a diet can be provided in an artificial diet formulated to meet particular nutritional requirements for maintaining a pest on such diet. The diet may be supplemented with a pest-controlling amount of an RNA that has been purified from a separate expression system to determine a pest-controlling amount of RNA composition or to determine extent of suppressive activity upon ingestion of the supplemented diet by the pest. The diet can also be a recombinant cell transformed with a DNA sequence constructed for expression of the agent, the RNA, or the gene suppression agent. Upon ingestion of one or more such transformed cells by the pest, the desired phenotypic result is observed, indicating that the agent has functioned to inhibit the expression of a target nucleotide sequence that is within the cells of the pest. 0026 The gene targeted for suppression can encode an essential protein, the predicted function of which is selected from the group consisting of muscle formation, juvenile hormone regulation, ion regulation and transport, protein synthesis and transport, digestive enzyme synthesis, maintenance of cell membrane potential, amino acid biosynthesis, amino acid degradation, sperm formation, pheromone synthesis, pheromone sensing, and any formation, wing formation, leg formation, development and differentiation, egg formation, larval maturation, digestive enzyme formation, hemolymph synthesis, hemolymph maintenance, neurotransmission, cell division, energy metabolism, respiration, an unknown function, and apoptosis. 0027 Another aspect of the present invention also provides methods for improving the yield of a crop produced from a crop plant subjected to insect pest infestation. Said method comprising the steps of A. Introducing a polynucleotide comprising a sequence selected from SEQID No:1 through SEQID No:906 or the complement or concatenator or fragment thereof into said crop plant, and B. Cultivating the crop plant to allow the expression of said polynucleotide, where an expression of the polynucleotide inhibits feeding by insect pests and loss of yield due to pest infestation. 0028 In certain embodiments, the expression of the polynucleotide produces an RNA molecule that suppresses at least a first target gene in an insect pest that has ingested a portion of said crop plant, wherein the target gene performs at least one essential function selected from the group consisting of feeding by the pest, viability of the pest, pest cell apoptosis, differentiation and development of the pest or any pest cell, sexual reproduction by the pest, muscle formation, muscle twitching, muscle contraction, juvenile hormone formation and / or reduction, juvenile hormone regulation, ion regulation and transport, maintenance of cell membrane potential, amino acid biosynthesis, amino acid degradation, sperm formation, pheromone synthesis, pheromone sensing, and any formation, wing formation, leg formation, egg formation, larval maturation, digestive enzyme formation, hemolymph synthesis, hemolymph maintenance, neurotransmission, larval stage transition, pupation, emergence from pupation, cell division, energy metabolism, respiration, cytoskeletal structure synthesis and maintenance, nucleotide metabolism, nitrogen metabolism, water use, water retention, and sensory perception. 0029 In other embodiments, the insect pest is a corn rootworm pest selected from the group consisting of Diabrotica undecimpunctata howardi southern corn rootworm SCR Diabrotica virgifera western corn rootworm WCR Diabrotica barber northern corn rootworm NCR Diabrotica virgifera Mexican corn rootworm MCR Diabrotica baltirda Brazilian corn rootworm BZR Diabrotica virgula Brazilian corn rootworm BZR and Diabrotica speciosa Brazilian corn rootworm BZR. 0030 Methods for improving the drought tolerance of a crop produced from a crop plant subjected to insect pest infestation. Said method comprising the steps of A. Introducing a polynucleotide sequence selected from SEQID No through SEQID No:906 or a fragment thereof into said crop plant, and B. Cultivating the crop plant to allow the expression of said polynucleotide, where an expression of the polynucleotide inhibits feeding by insects, pests and loss of drought tolerance due to pest infestation, are also provided. 0031 Yet another aspect of the invention further provides agronomically and commercially important products and / or compositions of matter including, but not limited to, animal feed, commodities, products and byproducts that are intended for use as food for human consumption or for use in compositions and commodities that are intended for human consumption including but not limited to corn flour, cornmeal, corn syrup, corn oil, corn starch, popcorn, corn cakes, cereals, and the like. Such compositions may be defined as containing detectable amounts of a nucleotide sequence set forth herein, and thus are also diagnostic for any transgenic event containing such nucleotide sequences. These products are useful at least because they are likely to be derived from crops propagated with fewer pesticides and organophosphates as a result of their incorporation of the nucleotides of the present invention for controlling the infestation of coleopteran pests in plants. Such commodities and commodity products can be produced from seed produced from a transgenic plant, wherein the transgenic plant expresses RNA from one or more contiguous nucleotides of the present invention or nucleotides of one or more coleopteran pests and the complements thereof. Such commodities and commodity products may also be useful in controlling coleopteran pests of such commodity and commodity products, such as for example, control of flower weevils, because of the presence in the commodity or commodity product of the pest gene suppressive RNA expressed from a gene sequence as set forth in the present invention. 003 to the method of producing such a commodity product comprising obtaining a plant transformed with a polynucleotide comprising a sequence selected from the group consisting of SEQID No:1 through SEQID No:906, or a concatenator or fragment or complement thereof, and preparing a commodity product from the plant or part thereof is also provided. Further, the method of producing food or feed, comprising obtaining a plant transformed with a polynucleotide selected from the group consisting of SEQID No through SEQID No:906 or a fragment or complement thereof, and preparing food or feed from said plant or part thereof is yet another aspect of the invention. 0033 The invention also provides a computer-readable medium having recorded therein one or more of the nucleotide sequences as set forth in SEQID No:1 through SEQID No:906, or complements thereof, for use in a number of computer-based applications, including but not limited to DNA identity and similarity searching, protein identity and similarity searching, transcription profiling characterizations, comparisons between genomes, and artificial hybridization analyses. Brief description of the figures 003 for Figure 1, bioassay of F1 corn plant events transformed with Pman 98503 SEQID No:820 and challenged with Western corn rootworm WCR. Figure 2, bioassay of F1 corn plant events transformed with Pman 98504 comprising concatenator C1 SEQID No:821 and challenged with Western corn rootworm WCR. Figure 3, selection of DB49 and DB248 fragments and schematic design of DB49 DB248 concatenator C38. Figure 4, dsRNase F1 F13 synthesized based on concatenator C38. Figure 5, DB49 DB248 concatenator 38 dose response fragments F1, F6. Figure 6, DB49 DB248 concatenator 38 dose response fragments F7 F10. Figure 7, DB49 DB248 concatenator 38 dose response fragments F11 F13. Detailed description of the invention 0035 The following is a detailed description of the invention provided to aid those skilled in the art in practicing the present invention. Those of ordinary skill in the art may make modifications and variations in the embodiments described herein without departing from the spirit or scope of the present invention. 0036 The present invention provides methods and compositions for genetic control of pest infestations. For example, the present invention provides recombinant DNA technologies to post-transcriptionally repress or inhibit expression of a target coding sequence in the cell of a pest to provide a pest protective effect by feeding to the pest one or more double-stranded or small interfering ribonucleic acid RNA molecules transcribed from all or a portion of a target coding sequence, thereby controlling the infestation. Therefore, the present invention relates to sequence-specific inhibition of expression of coding sequences using double-stranded RNA dsRNA including small interfering RNA to achieve the intended levels of pest control. 0037 Isolated and substantially purified nucleic acid molecules including but not limited to non-naturally occurrent nucleotide sequences and recombinant DNA constructs for transcribing dsRNA molecules of the present invention are provided that suppress or inhibit the expression of an endogenous coding sequence or a target coding sequence in the pest. When introduced thereto, transgenic plants that contain nucleotide sequences encoding the isolated and substantially purified nucleic acid molecules and the non-naturally occurrent recombinant DNA constructs for transcribing the dsRNA molecules for controlling plant pest infestations, and display resistance and or enhanced tolerance to the insect infestations, are also provided. Compositions containing the dsRNA nucleotide sequences of the present invention for use in topical applications onto plants or onto animals or into the environment of an animal to achieve the elimination or reduction of pest infestation are also described. 0038 The inventors have herein discovered that Contrary to the teachings in the prior art, feeding a composition containing double-stranded RNA molecules consisting of sequences found within one or more expressed nucleotide sequences of a Coleopteran species to the species from which the nucleotide sequences were obtained results in the inhibition of one or more biological functions within the Coleopteran species. Particularly, the inventors have discovered that feeding the double-stranded RNA molecules described herein to crop pest species such as corn rootworms results in the death or inhibition of development and differentiation of insect pests that ingest these compositions. 0039 The inventors have identified the nucleotide sequences described herein as providing plant protective effects against Coleopteran pest species. Amino acid sequences encoded by the cDNA sequences have been deduced and compared to known amino acid sequences. Many of the sequences are predicted to encode proteins that have some annotation information associated with them. The annotation information that is associated with a particular nucleotide sequence and protein sequence encoded therefrom is based on homology or similarity between the amino acid sequences deduced through translation of the coding sequences described herein as set forth and amino acid sequences that are known in the RN publicly available databases. 0040 cDNA sequences encoding proteins or parts of proteins essential for survival, such as amino acid sequences involved in various metabolic or catabolic biochemical pathways, cell division, reproduction, energy metabolism, digestion, neurological function and the like were selected for use in preparing double-stranded RNA molecules that were provided in the diet of Coleopteran pests. As described herein, ingestion by a target pest of compositions containing one or more desogenase, at least one segment of which corresponds to at least a substantially identical segment of RNA produced in the cells of the target pest, resulted in death, stunting, or other inhibition of the target pest. These results indicated that a nucleotide sequence, either DNA or RNA, derived from a Coleopteran pest can be used to construct plant cells resistant to infestation by the pest. The pest host, for example, can be transformed to contain one or more of the nucleotide sequences derived from the Coleopteran pest. The nucleotide sequence transformed into the pest host or symbiont may encode one or more RNAs that form into a desogenate sequence in the cells or biological fluids within the transformed host or symbiont, thus making the desoRNA available in the diet of the pest.